Good morning, students. Today we are going to start another cycle, that is Pentose phosphate pathway. Till the last class, we covered up to glycolysis. And this Pentose phosphate pathway also is a pathway for metabolizing, breaking down the glucose and to liberate energy-rich molecules like NADPH, FADH2, like that. Okay, so in that way, in that sense, we can tell that this pentose phosphate pathway is an alternative pathway for glucose metabolism. Okay, then there are some other names also there for this pathway apart from this hexose monophosphate shunt. So, which are the names? Phosphogluconate pathway. If for your university examination, if question came asking to write about phosphogluconate pathway or Warburg-Lehmann-Dickens pathway, all these are regarding this pentose phosphate pathway. Next. Is where it is actually occurring, taking place. This pentose phosphate pathway is taking place in the cytosol of tissues like adipose tissue, mammary gland, and also adrenal cortex. All these tissues, actually, this pentose phosphate pathway is taking place. Then, what is the nature of chemical reactions of this pentose phosphate pathway? While we are looking through that pathway, we can see that three type of reactions are mainly happening along that pathway. In that sense, mainly we can divide that pathway into first part and a second part. That first part consists of a series of oxidation reaction. Oxidation reaction, very simply we have to understand it as oxidation means we are taking energy from something, and in the place of reduction, that means we are giving energy. In this basic way you have to understand and keep it in mind while reading or looking through the any oxidation type or reduction type reactions. Okay. In this pentose phosphate pathway, the first part consists of series of oxidation reactions. And also this oxidation reaction results in the synthesis of energy-rich molecules like NADPH and 5-carbon sugars. Since 5-carbon sugars are produced in this cycle, in this path, so not a cycle, in this pathway is called pentose phosphate pathway because 5-carbon sugars are forming here. And the second part consists of involves a series of non-oxidative reaction. It's not taking or generating any energy-rich molecule. Instead, some reshuffling of the carbon skeletons of the compounds is happening in the in the second part of that cycle. These are the two parts. First part, series of oxidation. Second part, series of reshuffling. And even if oxidation of glucose here is the main thing that is happening in this cycle, we cannot say that this pentose phosphate pathway is a catabolic type. Instead of that, it is an anabolic. Why? How can we call it as an anabolic even if it breaks down the glucose? Because it donates, contribute so many intermediates required for some anabolic or building a process. That is because this pentose phosphate pathway, we can call it as an anabolic pathway. The major role of this pentose phosphate pathway, what are its major role? It generates the major portion of cytoplasmic NADPH. One major role is that to contribute the major portion of the energy-rich molecule called NADPH, and also to generate ribose 5-phosphate. What is the significance of this ribose 5-phosphate? It is necessary for nucleotide synthesis. Then what is the significance of this nucleotide? Nucleotide are actually building blocks of nucleic acids like RNA and DNA. So in that sense, we can very well understand the significance of this ribose 5-phosphate. So since the ribose 5-phosphate is contributed through this pentose phosphate pathway, it is that much important. Okay. That's a major role of pentose phosphate pathway. Then common intermediates. There are some substrates, some intermediates that present in this pathway is common to both glycolysis and also for pentose phosphate pathway. So that we are going to see which are that common intermediates present in both glycolysis and pentose phosphate pathway. They are glucose 6 phosphate, glyceraldehyde 3 phosphate, and fructose 6 phosphate. All these three compounds are common present in the cycles, in these two cycles, which are the cycles glycolysis and pentose phosphate pathway. So they can interconnect it because if pentose phosphate pathway proceed and after reaches the glyceraldehyde 3 phosphate or glucose 6 phosphate or fructose 6 phosphate, it can then redirect and can proceed like a glycolytic pathway. That's, these two pathway can interconnect at whatever point where all these common intermediates come. Okay. So in that sense, we can tell that these pathways are interconnected. So we can go and see, we can start to understand the major happenings that occurs in this pathway. This pathway starts with the glucose 6 phosphate, that itself a common substrate that present in glycolytic pathway also. Anyway, what is going to happen? We are going to understand about what is going to happen for this glucose 6 phosphate. In the beginning of this pathway, I have already told you that the pathway we can divide into two parts. First part consists of series of oxidation, second part a series of reshuffling. So we are going to see the first series of oxidation reaction. Three-step oxidation process are occurring in the first part. So glucose 6 phosphate. First oxidation is going to happen for this glucose 6 phosphate. As we know, oxidation means taking off electrons or protons, H plus. And that H is given to NADP. That way it get reduced and it become NADPH. The enzyme that helping the reaction, facilitating that reaction is glucose 6 phosphate dehydrogenase. Very easy to memorize the name of that enzyme. How? Its first half comes from the substrate's name itself. Glucose 6 phosphate. Then the remaining, as hydrogen is removing, we can in a sense, we can memorize it as dehydrogenase. D means removing, hydrogenase. So hydrogen is removing. In that way, for memorizing it, we can think it in that way. Glucose 6 phosphate dehydrogenase. First oxidation is completed and the product, what is formed out of that oxidation step? 6-phosphogluconolactone is formed. Okay. Then one oxidation finish. Then we are going to see next round of oxidation happening to this intermediate. 6-phosphogluconolactate. This 6-phosphogluconolactate again undergo oxidation with the help of the enzyme lactonase and form what? 6-phosphogluconate. Again, we are going to see one more round of oxidation with the help of another enzyme. Its name also very easy to memorize. In what way? 6-phosphogluconate dehydrogenase. Okay. And what is forming there? In this pathway, again one more NADPH is formed with the help of this oxidation. And the product formed here is a 5-carbon sugar, that is ribulose 5-phosphate. In the beginning itself, I told that it's a compound that is very necessary for the biosynthesis of nucleotides. Leave that. Then we are going to see what is happening to this ribulose 5-phosphate. So that oxidation series is finished by this step. Then we are going to see other reactions like rearrangement, reshuffling. Before that, we need to understand that this ribulose 5-phosphate can proceed either of two pathways. It can undergo, it can proceed through either of these two pathways. It can either proceed through this pathway or can proceed through this pathway based upon what enzyme is acting on it first. Okay. Then take. First we can consider this pathway. If ribulose 5-phosphate is acted upon by phosphopendose isomerase, that part you need to give more attention. Isomerase, if that enzyme is active, what enzyme? Phosphopendose isomerase is acting on the ribulose 5-phosphate. What may happen? This is the enzyme helping for isomerization. Isomerase. Give an isomerase form of this intermediate. And what is formed? Ribose 5-phosphate. From ribulose, ribose 5-phosphate is formed upon isomerization with the help of phosphopendose isomerase. Then it is going to have two steps of rearrangements. The first step of rearrangement we are going to see. Rearrangement is done by another enzyme, transketolase. That part also we need to give more attention. Ketolase. First set of rearrangement is always done by ketolase, transketolase enzyme. And what is forming here? Pseudoheptulose 7-phosphate is formed here. Then another round of rearrangement also happening to this pseudoheptulose 7-phosphate. The pseudoheptulose 7-phosphate again undergo one more round of rearrangement with the help of another enzyme called not ketolase, aldolase, transaldolase, and result in the formation of fructose 6-phosphate. This fructose 6-phosphate either can continue through the glycolytic pathway and results in again production of energy rich molecules. So in that way it can either connected to glycolysis. Okay. Then or either this can be changed into isomerase into glucose 6-phosphate and can re-enter the pentose phosphate pathway once more. Whatever it be. Then we need to consider about the other pathway. The ribulose 5-phosphate is acted upon by another enzyme called phosphopendose 3 epimerase. Epimerase. Here it is isomerase. If it is epimerase, then that isomerization result in the production of cellulose 5-phosphate, another intermediate. Then the cellulose 5-phosphate is going to have two rounds of rearrangement. First rearrangement, as we always know, that is done by a ketolase, transketolase. And what is forming here? Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, again a common intermediate present in both the glycolytic pathway and pentose phosphate pathway. Then second round of rearrangement. This glyceraldehyde 3-phosphate again undergo a second round of rearrangement with another enzyme, transaldolase, and results in the formation of erythrose 4-phosphate. And here in this pathway, there is one more chance of rearrangement is there. That rearrangement can happen only when this erythrose 4-phosphate combines with one more cellulose 5-phosphate. This one more additional cellulose phosphate is contributed by another round of this pentophosphate pathway. Okay, by that way, this second cellulose 5-phosphate combines with erythrose 4-phosphate and can undergo a rearrangement with the help of transketolase enzyme and results in the formation of two substrates, two compounds, glyceraldehyde 3-phosphate and fructose 6-phosphate, again a common intermediate of glycolytic cycle and also pentose phosphate pathway. So these compounds either they can proceed through glycolytic pathway once more and results in the generation of energy rich compound, or can proceed back to the pentose phosphate pathway itself. How it can proceed? Fructose 6-phosphate can directly isomerase to become glucose 6-phosphate and can start pentose phosphate pathway. Glyceraldehyde 3-phosphate either can continue down through the glycolytic pathway, glycolysis. In that way also it can generate energy rich molecule. That's about the pentose phosphate pathway. I think it's very clear for you. Then we are going to see the biological significance. Significance we already known to certain extent by this time. Anyway, once more we are going to see what is the significance of pentose phosphate pathway. It donates the ribose 5-phosphate. As we know, it is an essential building block, essential component for the synthesis of nucleotide. In that way, it is significant. And also it provides intermediates for glycolysis and also provide NADPH for fatty acid synthesis. In the fatty acid synthesis, NADPH acts as a reducing agent. So in that sense, this pentose phosphate pathway gives that reducing agent for the synthesis of fatty acid. Then glyceraldehyde 3-phosphate again can form and it can re-enter in the glycolysis. Next, we are going to see the last one of this last phase of the carbohydrate metabolism. It's biological oxidation. Now, listen to that term itself. Oxidation reaction that occur in living cells is called biological oxidation. Most probably, it happens in mitochondria. And it occurs in step-by-step manner. And the energy released in each step is utilized for various activities of cells. In biological oxidation, usually the energy releasing and energy consuming reactions are coupled. Are coupled. They are connected. Then we are going to understand about the transport of materials across the mitochondrial membrane. Some of these metabolite intermediates like pyruvate, all these are now present outside the mitochondria in the cytosol. So all these things we need to carry towards the inner matrix of the mitochondria. Then only we can proceed, we can continue the further breakdown of these metabolites and to convert it to energy rich molecules like ATP. So that transport is the next thing we have to understand. I will give you some instances, situation. In glycolysis, so many NADH are produced. And that that NADH are energy-rich molecules and it bounds so many electrons. That electrons, that NADH need to be transported towards the inside the mitochondria, first instance. And second instance also there, carbohydrate and fatty acid that break down in metabolic intermediates also need to reach the matrix of mitochondria, another instance. And also that after this complete oxidation, that energy rich molecule means ATP that synthesized inside the mitochondria also need to be transported out for the cellular, different cellular requirement, because the ATP is actually the energy currency for all our activity and it is producing inside the mitochondria mainly. And ATP as it is synthesized in the mitochondria is not to be kept inside the mitochondria. It have to be distributed out. It have to be transport out. All these things are important. If it is transport out, then only it can be taken up by the cells for its activities. So we are going to see the mitochondrial electron transport. This electron transport actually ends up in the final synthesis of ATP. The synthesis of this ATP is the ultimate goal of whatever metabolic process that's happening, that metabolic process that up to here we learn, because so many cycles we have learned regarding the breakdown of glucose, breakdown of carbohydrate, and all these will become will reach its end after it finish all the process that happening in the mitochondria. So we need to understand, we need to have a basic understanding regarding the electron transport. That's what I am going to give you now. Through many cycles, some pyruvate fatty acids are formed. So in a glycolytic cycle, we know the end product is pyruvate and it is present in the cytosol. That we need to transport towards the inner matrix of the mitochondria. That pyruvate and fatty acid end is transported towards the inner side and it again metabolized and converted to a 2-carbon compound called acetyl-CoA. This acetyl-CoA can then enter through the TCA cycle, that is actually happening inside the mitochondria itself. And as a result of TCA cycle, these products are formed inside the inside the matrix mitochondria. What products? 2 carbon dioxide, 3 NADH, 1 FADH2, 1 GTP, 3 hydrogen. And this time you may think that actually in TCA cycle 3 carbon dioxide are forming and 4 NADH are forming. Then why teacher is now telling about only 2 and 3? This means the first molecule of carbon dioxide and the first molecule of NADH. These two are formed. That formation actually happened outside the mitochondrial matrix during that link reaction. That's what is not mentioned here. And regarding all these molecules among them, this NADH and FADH2 carry high energy electrons. And these electrons need to be transported through the respiratory chain, and their transport result helps in the synthesis of ATP. How this transport help in the generation of ATP, that we are going to see now. How this electron transport through the electron carriers that is located in the inner mitochondrial membrane helps in the generation of ATP. How ATP is generated, that we are going to see. Then organization of electron transport chain also we need to understand how that electron transport chain. Electron carriers are arranged in the inner mitochondrial membrane. Mitochondria have outer membrane and inner mitochondrial membrane. And that electron carriers are arranged in the order of their redox potential. And 4 multi-subunit enzyme complexes are usually acts as the electron carriers. They are named as complex 1, 2, 3, 4 is name. I have given here. Complex 1 is called NADH dehydrogenase complex. Complex 2 is called succinate dehydrogenase. Is a enzyme complex that present that we already learned in glycolytic cycle itself. This enzyme helps in the conversion of succinate to fumarate. And that step actually that FADH2 is forming. Is complex 2. Then complex 3 is ubiquinone cytochrome oxidoreductase. And also then cytochrome oxidase. This complex 4. And as the electrons traverse through all these complexes, so much energy is liberated, and using that energy from the matrix of the mitochondria, protons, that means H plus, are translocated towards the inter mitochondrial space. Inter mitochondrial space means the space between the outer mitochondrial membrane and the inner mitochondrial membrane. We are going to see the process that happen associated with the complex 1. The complex 1, the electron electron pass to that complex 1. When this happens, if NADH become the electron donor, if the NADH acts as the electron donor, then that electron pass through the complex 1. 2 electrons, 2 protons, it is passing through this complex and transfer that electrons to ubiquinone molecule. And through this pathway, through this transport, so much energy is released, and that energy is called free energy. Using that free energy, some protons also exported from a matrix to the inner membrane space. That way, we can the mitochondria is able to build up a concentration gradient across the inner mitochondrial membrane. Start to develop a concentration gradient of proton. Next, complex 2. If that electron donor is FADH2, then that time that electron is passed through that complex 2. Whenever, in what situation complex 2 acts as a transporting center of electrons, whenever FADH2 acts as the electron donor. Then that electrons pass through this complex 2 and transfer that electrons to ubiquinon, another molecule of ubiquinon. And here there is no free energy. As there is no free energy, it's not possible to translocate any proton from the matrix to the intermembrane space here. Next, ubiquinon cytochrome oxidoreductase. That is complex 3. That ubiquinon that carry electrons from complex 1 and complex 2. They are translocating their electrons to another electron carrier called cytochrome C. That way, two cytochrome C are generated here. And here also some free energy is available, and using that free energy, protons are translocated to the intermembrane space that adds to that concentration gradient generated across the inner mitochondrial membrane with regard to the proton. Understand? Again, that concentration gradient increases. Next, cytochrome oxidase complex 4. Then what is happening in regarding to this complex 4? Complex 4 as a cytochrome C that carries that electrons and transport that electrons through complex 4 and finally transfer it to oxygen. And after receiving that electrons, oxygen take 2 more protons from the matrix of the mitochondria and convert to one molecule of water. That water we are actually removing through our sweat, our urine. Okay. And that electron transport is come to an end by this reaction. That the reaction means by the formation of water. Then we want to know how this ATP is forming. This electron transport is everything come to an end. And we think that as a result of the electron transport, mitochondria have generated a concentration gradient of proton across the inner mitochondrial membrane. That is important. That is playing the major role for the generation of ATP. For that, okay, let me see that. Before finishing that, before going the remaining part, we need to understand about oxidative phosphorylation. Oxidative phosphorylation chemi-osmotic hypothesis, very important, has asked for so many times for the examination. It is proposed by Peter Mitchell, 1961. I want to learn about what's that hypothesis. This is the inner mitochondrial membrane, and these are the electron carriers, complex 1, 2, 3, 4. And this is the ATP synthase. As the electron pass through these electron carriers and finally give to oxygen and one molecule of water is formed here. And also along with this, some amount of energy release in each step, and as a result of that, a proton gradient has built up across the inner mitochondrial membrane up to where we have understood that. Then what is happening next? Using then that proton gradient, proton is more in this area now. That more amount of proton. What its tendency? It always has the tendency to move from a region of higher concentration to a region of low concentration. Now, low concentration is less here and concentration is high here. So, what these protons? They are moving to matrix through this ATP synthase. In ATP synthase, this part contains a channel. Through that channel, it passes towards the matrix. It's that movement results in the rotation of the ATP synthase, and that rotation help in the release of the ATP already synthesized in the ATP inside the ATP synthase. In that way, that electron transport help to create proton gradient, and that proton gradient helps in the rotation of ATP synthase, and rotation of that ATP synthase help in the releasing of ATP. In that way, mitochondria synthesize the ATP, the energy currency. Okay, then we are going to see that proton is moving through that ATP synthase, and the ATP is released, liberated from the ATP synthase. In a single sentence, we can express chemiosmotic hypothesis is a coupling of electron transport with the phosphorylation of ADP. Coupling of electron transport with the phosphorylation of ADP. Then we are going to see about the ATP synthase. ATP synthase is an enzyme. We know it is synthesizes ATP using the energy from the backflow of hydrogen to the matrix. And where it's located, it spans the inner mitochondrial membrane. Then some other name also present for ATP. Where is the cursor? ATP synthase. What was the other name? Elementary particle or oxisome or F1, F0 ATPs. Three names are there other than ATP synthase. Which are the name? Elementary particle. Elementary particle. Elementary. Elementary particle or oxisomes or F1, F0 ATPs. Going to see the structure. For many times the ATP synthesis also asks for the examination. So you should learn all these things, and also you must know how to draw the structure also. The figure of ATP synthesis also you have to add for that answer whenever such question came. Structure, regarding that structure, it is a lollipop-like structure. It has two substructures are present. Then substructure means F1 portion is present and F0 portion is present, and stalk is present. And F0 is a barrel-like a transmembrane protein composed of 10 to 12 subunits. They are arranged in the form of a barrel, a channel, and in central portion have a passage also for the translocation of protons. And a stalk is present. Stalk and F1 consists of 5 different kinds of proteins. Stalk plus that F1 consists of 5 different kinds of proteins like alpha, beta, gamma, delta, and eta. And this is the site of ATP synthesis. This ATP synthesis is always called a reversible coupling device. Reversible coupling device means, reversible means backward. Forward reaction, what is the forward reaction? It's the synthesis of ATP from ADP. Then we need to think reversible means the conversion back of ATP to ADP again. That means breaking down of ATP. Breaking down of ATP always results in the liberation of so much energy. And using that energy again, the ATP synthase can translocate proton back to this intermembrane space. So in that sense, we can call ATP synthesis as a reversible coupling devices. Then there are two scientists, these persons actually first elucidate this structure, and they are awarded Nobel Prize also for this. They are called Paul Boyer and John Walker. Then we need to understand the peculiarity of outer and inner membrane of mitochondria. The outer membrane contains numerous porins. They allow the easy diffusion of molecules up to 10 kD molecular weight. And then what regard to inner membrane, it is rich in various proteins. This inner membrane is freely permeable to some molecules like oxygen, carbon dioxide, and water. And it shows some controlled permeability to ATP, ADP, pyruvate, calcium and phosphate. And this property is very significant because this property actually helping to generate the ionic gradient, concentration gradient of ions across the membrane. That about the peculiarity of inner and outer membrane. Then we are going to see another section, hormonal control, because by this way we finish all the pathways for carbohydrate metabolism. Now we need to how it is regulated, the carbohydrate metabolism. Mainly three hormones are significant. The three hormones we are going to see to control the carbohydrate metabolism are insulin, glucagon and epinephrine. Among this, only insulin its secretion happens whenever the blood glucose level becomes high. At that time, insulin is secreted. Other two hormones, glucagon and epinephrine, they are secreted when the blood glucose level comes down. Okay. Insulin means it's secreted when blood glucose level is high. Blood glucose level high means we need to take that excess glucose and to store it in the form of glycogen. That is understood there. So that duty may be done by this insulin. That duty may be prompted by the hormone insulin. Insulin promotes glycogenesis, means synthesis of glycogen. And in that way, the additional excess glucose can be stored in the liver or some other tissues in our body. And also it inhibits glycogenolysis. Glycogenolysis means breaking down of glycogen to form glucose. That pathway it inhibits. Some insulin responsive cells are present in our body. These cells have some receptors also in its outer membrane. These receptors bind the insulin molecule. This happens when the blood glucose level is high. And this binding results that glucose transport system. Glucose transport system means it says transport system exists there. That transport system actually transport the additional glucose that present in the blood towards the inside this cell. And then that glucose get converted and stored as glycogen. And altogether, what is the some net result? Glycogen synthase is activated. Glycogen synthase is the hormone responsible for the synthesis of glycogen. It is activated. And at the same time, glycogen phosphorylation is inhibited. Glycogen phosphorylase, what is its role? It prevents, actually, it is responsible for the breakdown of glycogen. As it is inhibited, that breakdown of glycogen is prevented. This is the net result of the presence of this insulin. In that way, it regulates the carbohydrate metabolism. Excess glucagon, it has secreted when blood glucose level is low. Then low means we need to synthesize, we need to get more free glucose for the proper functioning of the body. So what we need to know, we need to facilitate a glycogen breakdown and also to prevent the breaking down of any glucose molecule. Glucose molecule breaking down pathways we know glycolysis and pentose phosphate pathway are mainly glucose breaking down pathway. We can go and see. Glucagon stimulate gluconeogenesis. Gluconeogenesis means generation of glucose from non-carbohydrates substrate. And also it stimulates glycogenolysis, means breaking down of glycogen and to free glucose. And this glucagon acts by the way of G protein coupled signal pathway. This G protein coupled signal pathways, I think it's already known to you because you have learned again learned this the same topic in an endocrinology chapter and also in your last semester. I am just brush up your memory. What's happening here that whenever the blood glucose level is low, glucagon is secreted by the pancreas and that glucagon go and bind with that G protein coupled receptor. As a result, it undergoes a conformational change and the G protein associated with that receptor get activated. and its alpha subunit detach from it and goes and bind with the adenyl cyclase. This binding of this G alpha subunit of G protein activate this adenyl cyclase is called an effector enzyme. It become activated. This activated adenyl cyclase then results in the production of a second messenger called CAMP. CAMP is formed. This CAMP is activated by the phosphorylation using 1 ATP, and this activated CAMP in turn activate another protein kinase. That protein kinase is called CAMP dependent protein kinase. Is an enzyme, and its activation actually result in activation some other reaction over two other enzymes that we are going to see. Kinase means its action is to add phosphate on some other substrate and to regulate its activity. So in that way, CAMP dependent that protein kinase add phosphate to one protein called fructose bisphosphate and another enzyme called phosphofructokinase. By adding phosphate to this phosphofructokinase, its action gets suppressed. And also by adding phosphate to fructose bisphosphate, it become activated. And because of the suppression of phosphofructokinase, what is the end result? Glycolysis become suppressed because it's a key enzyme such supporting glycolysis. Glycolysis means breaking down of glucose. In that way, that glycolytic pathway can be stopped. In that way, as the glycolytic pathway is stopped, that means the glucose level can increase in the blood. And also, yeah, the fructose bisphosphate, it activator means it's an enzyme helping from gluconeogenesis. The gluconeogenesis means again additional glucose can be synthesized from non-carbohydrate substrate. This is the action of glucagon. It's mentioned here. Gluconeogenesis increase and glycolysis is suppressed. Next, we are going to see the role of another hormone called epinephrine. Epinephrine, what actually epinephrine also secreted when the time the blood glucose level is very low. Epinephrine, and it also acts by the way of G protein coupled pathway. Epinephrine bind that receptor G protein alpha subunit detached and bind adenine cyclase second and the effector enzyme. Second messenger CAMP get activated, and CAMP which in turn phosphorylate and activate another protein kinase that is called CAMP dependent protein kinase. And it for which in turn again phosphorylate another two enzymes like glycogen synthase and phosphorylase kinase. What these enzymes are you actually using? Glycogen synthase means it is converting the glucose and helps in the building up of glycogen that we actually need to stop. So that thing is happening here by suppressing the glycogen synthase. And by phosphorylating phosphorylase kinase, it get activated. This enzyme get activated. As it get activated, what the result occurs? Yeah, the glycogen phosphorylase, another enzyme. It activate another enzyme called glycogen phosphorylase. This glycogen phosphorylase is the enzyme responsible for the breaking down of glycogen. Breaking down of glycogen again free more free glucose, and in that way it can raise the level of glucose in the blood. These are the effect of these three hormones. These are three hormones are playing very important role in regulating the carbohydrate metabolism. The metabolism of other two biomolecules, that is lipids and proteins, taken by Elizabeth teacher later. So for me, this chapter is end, and also this today's section. Thank you.